Okay, so these are our highlights for our special senses, which is certainly a branch of our nervous system. We've been learning about nervous system already when we were talking about the organization of it. Remember that with our nervous system, we've got our central and peripheral nervous system, right? So we have central nervous system, which is going to be our main processing center, made up of the brain and the spinal cord. Then we have the peripheral nervous system, which is going to have an afferent division, which is leading into the central nervous system, relaying information about the current state of the world and our body to the central nervous system. And then if it has to make any adjustments or decisions about it, that is going to be going out the efferent division that we see here, which is subdivided into kind of a voluntary or somatic nervous system and an autonomic, or what we like to think of as the automatic nervous system instead. So what we're really going to be focusing on is over on this side here, where we're looking at sensory information coming in from around the world about us, or even from the world within us. Okay, so in terms of what you need to know, you certainly should know the diversity of of receptors, if you will, that we have in the body. Now remember that the receptors their main role is to translate a particular stimulus or modality into something that the nervous system can understand. Remember that the nervous system can only understand electrochemical signals, right? Those little waves, those action potentials or gradient potentials. And so these receptors change some particular stimulus into that so that the brain and the spinal cord can understand it. So you should know the diversity of different types of receptors and what they are transducing, what they are translating for the nervous system. Now we do go through some of the simpler ones, things like thermoreceptors and pain receptors, and even some of our touch receptors like our Meissner corpuscles that we find up in the superficial layers or the more superficial layers of the dermis, as well as things like Pacinian corpuscles, which are down deeper. But really, most of the chapter focuses on the different special sense organs, entire organs that are dedicated to translating this world around us. So we start out with vision. You got to understand what the stimulus is for vision. Of course, it's going to be photoreceptors sensing photons of light or light waves. And so we go through a little bit about what we call visible light. And then it's pure, like we've seen in other systems, anatomy and physiology of these organs. So you need to know the different parts of the eye and the role of each of those different parts. Ultimately, what we have is light coming in through here, right? It's going to first impact the cornea. It's going to pass through the anterior cavity. It's going to pass through the pupil. It's going to go through the lens, and it's going to project back to the retina. Now, depending on the angle of the light coming in, it may end up on different parts of the retina, to be sure. But a lot of the light and the image that we are able to create with our eye get focused back here on the fovea. And the fovea is an area of the retina, a very specialized area, that has all cones and no rods. It gives us very detailed vision, at least in highlight conditions. So you do need to know the two different photoreceptor types that we have, the fact that we have rods for low light vision, that we have cones for high light conditions, and we have three different types of cones. They both use a rhodopsin molecule, and you should know that that is the actual molecule that harnesses the stimulus, the light stimulus, and starts to change it into an electrochemical signal. So that's what we were talking about there. But also, like we said, the fact that you have three different cone types. So you should know that we have blue cones, green cones, and red cones. And that when we combine the stimulation to those three different cone types, then we're able to discriminate millions of different colors. Of course, that's nowhere near what the mantis shrimp can discriminate, but that's another story. Then once we get that stimulation to the photoreceptors on the back of the eye right here, knowing the pathway, if we were to trace the reception of light into where it's perceived in the brain, it would have to pass through, of course, the optic nerve, through this X that we call the optic chiasma, route it through the thalamus, and then back to the occipital lobe, because it's in these occipital lobes where vision is mainly processed in the brain. Okay, So then we do also spend a lot of time on the ear. So the eye and the ear are the bulk of the information that we find in this special senses chapter. And we need to know, again, much like the eye, all of the different structures of the ear. We need to know whether they make up the external ear, the middle ear, or the inner ear, and what their particular role are in, again, transducing a signal. In this case, the signal is going to be sound waves. Okay, so again, you need to know the different parts. We can see the parts of the middle ear here, how the ear ossicles are translating and even amplifying the signal to the cochlea. And inside the cochlea is where we actually have the receptors. And these receptors are hair cells that are physically going to be bent. In terms of the receptor type, they are mechanoreceptors. So when a sound wave comes in and causes this basilar membrane to vibrate, these hair cells are going to bend as the whole spiral organ kind of rams into the tectorial membrane, the ceiling that's over it that we see here. And then those hair cells will send signals into the brain saying that we have heard something. Okay, now the ear counts as special sense organ in terms of hearing, but also in terms of equilibrium. There's two types of equilibrium. There's called rotational or dynamic equilibrium, and it has to do with these semicircular canals that extend off in three different directions, the X, Y, and Z directions. There's also the utricle and the saccule. These are found in the vestibule, and these are associated with what's called linear equilibrium or static equilibrium, because it doesn't require movement to get these to do their job. This involves, again, hair cells, like we see here, just like we saw in the semicircular canals and in the cochlea. And now what we have is the presence of otoliths that will pile on top of certain hair cells, and that will cause them to bend and send a stimulus in. Then, of course, we go through taste and we go through smell. Both of those are chemoreceptors, but you should know the specific chemicals that are able to stimulate them and kind of where those receptors are found. And that's really it for special senses. So, while those organs are somewhat complex, at least it's not a whole, if you will, organ system like we've seen with a lot of our other chapters as we go through them.